day life, remember? And it jiggles. Essentially just jiggles. And then you gotta make something out of that because this actually took like 3, 4, 5 months to generate this jiggling. And you gotta ask yourself, what am I actually learning from this jiggling? And so Python comes in. Python comes into play to analyze this jiggling in a quantitative way. So I mean, it's not, computer models don't exist in a vacuum. I've got years of data to draw on, experimental data, I should say. For example, the discovery of this protein was in 2003. It actually won a Nobel Prize, this puppy here, so it's pretty important. I mean, I could elaborate on why it's more important, like what kind of diseases result in problems with this protein, but we won't get too deep into that. Anyway, this jiggling is interesting because there are certain parts of the protein that are more important than others, I would say. So I kind of hid the protein there. I'm showing you just one particular amino acid. You guys know amino acids, probably know it on a sort of a general sense, because when you consume protein, chicken breast, protein powder, you're basically eating these things. Your body needs them to make new proteins. But this is one in particular that I'm interested in. It's called arginine. So arginine's important in this protein. So what I'm going to do is use Python to extract out what's going on with arginine in kind of a geometric fashion. Because again, this is not bioinformatics, this isn't sequences. This is a study of biology in a purely structural way. Like, this is a real three-dimensional object, and my raw data, the data file I've generated, is just the positions of atoms in space versus time. So I have a time series, just like people in finance have a time series of stocks. I have a time series of, the position of this atom over time, and it is, it's essentially, I can use the same tools. I can leverage all those finance folks for the power of science. And so let's dig in. As you can see, already just looking at this particular one, it kind of adopts a flattened out state and then an up-facing state. They're kind of like two different states going on. And I'm showing even 4 of them because your body actually has 4 of them in a cluster. 4 of them actually look like this. So there's all 4 of them are here. I gotta analyze all of them and kind of do it in sort of a statistical way so I can, kind of learn about it in a general average sense. It's difficult because I see something happen and I like to sit back and watch movies. I'll say that. I just love, good part of the job. It's very visual. I can sit back and watch this, but again, quantitative aspects I need. So let's dig in. This is an IPython notebook that I prepared. And there's some little snapshots of this arginine in its sideways state and in its up state. And sideways state and up state kind of vaguely doesn't mean anything to you, but let's just say that one of them could block water flow and one of them couldn't. So maybe if it was in an up state, water should flow nicely through, and then in a down state, water couldn't. So that would be important for a protein that's designed to make water flow. Anyway, there's very little that's like my field specific in this IPython notebook, except at the beginning when I'm pulling out the data. So there's one library written in Python that's called MD analysis, and I'm going to use it to extract out data about this sequence of amino acids. Wow, gonna have to increase font size here. That's easy. Okay, so I'm going to extract out this thing called the dihedral angle. You can imagine my Egyptian pose is like a sequence of 4 atoms where my fingertips is one atom, and my wrist is the other, my elbow is one, and then my shoulder is the other. And so it's this angle that I'm extracting, just based on, I don't know, this is like a geometric thing. So I'm extracting that as for my protein, as one of my, I guess, features, or like, pieces of kind of intermediate data. It's not the raw data, but it's like a little more meaningful because I can grasp it mentally. Anyway, extract out that puppy, which is really just a loop over this like binary data file, blah, 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 blah. You get a data frame. I throw it into a data frame, let's say, where the index is the time. And remember, this is time on a time scale of like picoseconds. So it's like extremely small times, but it's real time nonetheless. It's like, I don't have the exact timestamp. I can use a date time because it's just, I probably could though. I don't know how many decimal places that goes to. But anyway, stick to the indexing for normal, for now. we'll extract out those properties, those angles. I call them chi1 and chi2. And I also extract out something a little more tangible, which is like just the distance from end to end. I just kind of take this atom and subtract this atom, and then it kind of gives you a sense of this downstate or upstate. So those are my three things, chi1, chi2, and z. So you throw it into a data frame, and you can sort of do the data frame, too many numbers, data frame describe is a little too vague even for me. It's like, okay, I have a mean value of these angles, and I also have a mean value of this z, but it just doesn't do much. We've got to plot it to really get a sense of what we're looking at here. So I scroll down a little bit. There's some stuff you don't need to see. But the chi1, chi2 space is here. Chi1 and chi2, this is the angular space. So they basically there's a lot of basins. The data is kind of scattered across a lot. I use the Seaborn library. It's nice plotting library because I can get a little projection on either axis. So this is the chi1 data projected here, and this is the chi2 data here. So anyway, these are actual like regions which biology people understand. They actually have names, like these quadrants, I would say. GG, G+, TT. And you can see most of the data is actually here, 26% of the data. I kind of did a cool thing, like a bit of fast code to count the populations. Anyway, group by, I gave them all basins. I assigned them all basins based on these like slices I just showed you. So I named them all, and then I did a data frame, like with pandas, you can do a group by, and I group by that, just that quantity, that column of the data, and you can like instantly sort of summarize the populations of this, of these regions of space. But again, what does that tell you? It tells you that most of my data is clustered in here, but what about here? What is this, like what does it mean? So you can also take this Z quantity, which is getting a little more tangible, plotting the Z values, but I'm going to plot it into the chi1, chi2 space. So the same plot, but instead of doing a histogram, this is really just a histogram, right? Like I have a piece of data, time, whatever, it fits into this bin. But what if instead of showing population, I took another quantity like Z, and I kind of mapped it onto this span. So I said, anytime there's an angle here and here, instead of writing out the population of that, I'm going to write out the Z value, the mean Z value. Again, super nice with pandas. Wish I could elaborate a little bit more, but this is short lightning talk. Anyway, I like to help on this one because actually I think it could be done better because I iterate over rows in the pandas data frame, which is like not recommended. But anyway, let's look at the result. So as you can see now, there's like some color to the whole picture. We know that these most populated basins are here and here, and those correspond to Z values that are like 0 or negative. So that's really the Z value that's horizontal. So now we understand that most of the data is like horizontal, but there's some interesting data like over here, I should say it's periodic, angles are periodic, so actually it wraps around. So this basin here does the same. This doesn't tell you that, but it's the same basin. And it's also really interesting because it's always kind of pointed up. It's a super upstate. So that's interesting because like you do a computer simulation, it's really jiggling, all these sort of things happen. What do you tell somebody who's studying this protein, like, the protein does everything, the protein does so much. It's not just one picture, it's like a lot going on. Anyway, there are things you can measure experimentally. Experimentally, like, again, the computer, no one really trusts you in the computer modeling world. And they say, you showed me this plot, but is it real? Like, what is it, how do I trust you? So there are experimental things you can measure about proteins. For example, like you could take a beaker full of protein and you could see, like by applying these complicated magnetic fields, you could see which things are exposed to the water, exposed to the solvent. And so that would be an NMR measurement. And so that's what our collaborators did. And they did NMR measurements and they said, Chris, I know a few amino acids in this protein that should be exposed to the water. Do you also see that in computer simulations? And I say, okay, well, I'm going to need to build another data frame. So I'm extracted something called hydrogen bonding. So hydrogen bonding is what makes water so watery and nice. it's this interaction between an oxygen and a hydrogen, and it makes like water form this sort of continuous like network, this hydrogen bonding network. But water's not the only thing that hydrogen bonds. Proteins also do it too. And that's probably why the protein flopped over, because it wouldn't just choose to do that randomly. It actually had a partner over there that I didn't show you. It had like a buddy of some kind that it was interacting with. So, another data extraction that's like specific to my field. This is another cell that's just my field related. Do that, and you get another time series, another data frame and time series where it's like at different time steps, there are many different people you're talking to, people you're making a hydrogen bond with. I'll just show you an example to give you some tangible things to think about. Oh yeah, this will do it. So here I showed you that arginine before, but these are some neighboring amino acids that this arginine might talk to. Again, real computer simulation. So I'm going to put it on its side, and sometimes it's talking to this guy above, and sometimes it's talking to this guy over here. So I got to quantify that again. So that's the reason I built this data frame, and you can pull it all together. So it's one thing, I can extract out the populations of who you're talking to. You're talking up, you're talking left, you're talking right. And then you can join it together with what the data I previously showed you. And I can extract out who are you bonding with in those Chi2 regions. So let's do that, and this is just a bar chart that kind of summarizes. My bar chart broke. Oh yeah, just scrolled crazy. Okay. Our chart shows that there are random basins. These are the G basins. Sorry, I don't have the previous plot. I can switch back to the previous plot. But there are some of those basins where you're talking to someone who's above you and someone who's beside you. Sometimes not. So I kind of if you had data you didn't understand that you didn't have a physical intuition about, you might do a lot of complicated machine learning. But I can leverage my intuition about geometry and stuff like that in order to make sort of a more, directed study, directed scientific study. So I make, I separated out my data set into a way that made sense for my brain. I didn't use an unsupervised approach to do that. Anyway, given this, it doesn't seem like much, but it's encapsulates sort of a thread of research, which is leading us to that comparison with experimentalists. So experimentalists, as I said, they apply complicated magnetic fields, and they can study, signal intensity is the units of this graph, and this is some complicated, thing. But we can compare directly their observations in beakers with the hydrogen bonds that I got up there. So there's reasons why they don't have the exact same numbers as I do. They have, for example, they have N127. And I also found that in 16% of one of the basins, I had hydrogen bonds with N127. So, okay, 16%, that's not much. But the best my data set can do is say that this particular basin best represents the experimental data. So, it's like I narrowed in on the juiciest gem of my data set. And I can come to my collaborators and say, your data agrees with this subset of my massive mess of data. And now you can take a look at this one. I would say, hey collaborators, now I'll show you pictures of that, and you can see if it matches other things. And you can really, start to build a story of how this protein works, how it could malfunction, and Python brought us there to do that. So, I know that's pretty fast. Didn't elaborate on some of the cool-ass pandas I did, but I'll just say, that's sort of the approach I'm going for. Let me make one more comment. Just a general comment is that it took supercomputers to generate the raw data. But, we're not really at the big data stages because I get a rich data set where each time step has so much information in it, but I really don't have billions of data points. In a sense, I have billions of atoms, but I really, in time, the number of time steps is not that much. But we're working towards that, and people in my field are working towards that. I'm not the only one, but it's a burgeoning field, at least in the structural biology sense. Bioinformatics people are a little bit ahead in the data science general sense, but this is a cool area also. So you get movies. Any, I don't know, did lightning cost any questions? Maybe they did. Go ahead.
Yeah, I understand everything except why it's used for this. Oh, okay. So, are you? How long it took for you to come up with the whole code for analyzing the data that you got from that you have in the data set? So, how long did it took for you?
Yeah, it takes a day or two sit down in order to like make an IPython notebook like this. But once you start to doing it, you start to realize it's a big routine. It's like, it's just extracting another piece of data, not this amino acid, a different amino acid, so on and so forth. So yeah, really, I would say 3 days to make this notebook. And this is a subset of the data on my laptop, but it's really like, it takes like maybe a few hours to run this IPython notebook on the supercomputer with the full data set. Which is really fast. And yeah, sorry that was crazy. I'd like to give you more movies to watch. But we got other things